What's up, AP Bio Penguins? It is Friday, which means it is FRQ Friday, and it is also the last FRQ Friday. I know you are all super sad that I'm not going to do this every Friday for you, but today is going to be our last FRQ Friday. So we have 4 questions to go over today, and I didn't realize until I was setting it all up. I was like, oh no. These are the same questions that I did for marker learning. So if you've looked at my data analysis component of marker learning, then some of these are going to be repeats. There is some new stuff. I go through the entire question instead of just the data analysis part. But I do want to just forewarn you that I am, we have 3 different things that have to do with error bars today. Because I know that's a hot topic. We have to make sure we definitely know our error bars for the exam, whether it's a multiple choice question, whether it's a free response question. So I think I specifically designed this to be the last FRQ Friday so that you would see this error bar stuff right before your exam. And remember, by this time next week, you will be done with your AP exam. And if you decide to take the first administration for all your exams, then you will be done with all of your AP exams. So yes, way to go. I'm so proud of y'all. So keep on pushing. We're almost to that finish line. It's definitely not a marathon. I'm sorry. It's a marathon, right? It's not a sprint. You just got to keep working a little bit each day and you will rock it, right? So let's get started with today's FRQ Friday. So as I said, there's 4 different questions we're going to do today. We'll do 2017 number 1, 2016 number 8, 2015 number 5, and then 2014 number 4. Okay. So the first question is one of my favorite questions. I actually give this question to my students on the very first day of class. Not really the first day, but the first FRQ day that we have. So we usually start school on Monday, and then on Wednesday, I hand them this question. And so the reason why I hand them this question is because they know every part of this question without having taken biology. Okay. So sometimes you're going to find questions like this, right? You're going to come into that test and you're like, oh my God, I didn't have to know any biology for this, which is fine because they're assessing your science practices. They're assessing whether you know all the behind the scenes information. And my students take research before they come to me. So they've already done all that science practice stuff. So let's focus on our questions. So, and I did this really fast. So all my little underlinings aren't going to happen today. So in flowering plants, pollination is a process that leads to the fertilization of an egg and the production of seeds. In some flowers attract pollinators, such as bees, using visual and chemical cues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, and certain plants also produce caffeine in the nectar. Caffeine is a bitter tasting compound that can be toxic to insects at high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effect of 0.1 molar caffeine on bee behavior. The results are then shown in this table. So as you see in the table, you have our control, we have our caffeine, we have 10 minutes, and we have 24 hours. And it looks like that we're testing the memory. So it's about whether they kind of came back to the same source, right? So if they remembered it, they would have come back to the same flower. If they didn't remember it, then they were not going to come back to the same flower, right? So on the axis, and I already went ahead and labeled some of it, construct an appropriately labeled graph to illustrate the effect of caffeine on the probability of bees revisiting. Okay, so first thing you have to think to yourself is, what is an appropriate graph for this, right? I have 10 minutes and I have 24 hours. So what would be the appropriate type of graph that I would make? So you think to yourself and say, okay, well, I'm working with time. Time's always a line graph. Nope, not on this one, right? Because we have 10 minutes and we have 24 hours. You need to be able to have an equally spaced scale for your graph, right? So there's no way that you can equally space 10 minutes to 24 hours. That's such a huge jump. So you want to make a bar graph for this one. Okay, so you should have a bar graph. So now you could have done it one of 2 ways. Okay, you could put your controls together and you could put your caffeines together, or you could put your 10 minutes together and your 24 hours together. It all depends on you and your kind of thought process. You just had to make sure you had a key to show it all. Okay, my thought process is I'm going to show the times together. Okay, and so when I go ahead and I graph this, I went ahead and I put on the y-axis, I have my memory. Okay. And notice that I use the exact same thing that you see in the table. Like I literally have named it the exact same thing. You will not go wrong if you label it the exact same way because it already would have the units and everything else you have to have. And same thing down here. We have our 10 minutes, our 24 hours, and it looks like that I might have cut off the time part when I made my movement over, because I literally just copied it from Marco Learning's session. Okay. So we put in our grid. Okay. And again, you want to have your graph in there. And what am I missing? Notice here that there's this plus or minus next to each of those. What the heck is that? I don't even know what that means. That is your error bar. So if you ever see a plus or minus, that means you need to have an error bar on it. Okay. And so you're going to add that much to your mean, and you're going to subtract that much from your mean. Okay. So when we graph it, you're going to see right here. I thought I pressed the button. Yeah, I did. Okay. So when you graph it, we went up on our control. We went up from 0.72 up to, 0.09 above, and then 0.09 below. And then right here, we're going to go above and below by, what is that? That's our caffeine at 10 minutes. We went above and below 0.7 and 0.7. Okay. Again, I'm doing this on a computer, so it's not as accurate as it should be. So if you're sitting there going, Miss Jones, that's not exactly perfect. Y'all, I'm doing this on a computer. I think you can be okay with me. Okay. So then we do the same thing with our error bars for our 24 hours. So this is what your graph should look like, right? You should have your axes labeled. Okay. You should have it scaled appropriately, and you should have error bars on it. Notice I also have a key up here to tell me what my colors mean. Okay. You want to have some way to signify which one's your control and which one's your caffeine, since you were just labeling the bottom as 10 minutes and 24 hours. Okay. Notice on my scale though. Okay. Notice that I just have a huge gap down here. You do not have to label every single point. Okay. You can skip. For me, I believe I made it every 2 spaces is 0.1. Okay. So as long as every time you count up 2, you end up with the correct scaling, then you're perfectly fine. You can skip whatever numbers you want as long as you have appropriately labeled the ones that you did label. Okay. In addition, you do not have to start at 0, right? You can start at whatever number you want. Okay. And I usually tell you, it's always going to be a 20 by 20. And that's because I didn't super analyze the practice tests. Based on the practice exams, they may not be a 20 by 20 grid. So before you get started on your graph, go ahead and take a moment and count up how high you can go. Take into account your error bars. Okay. Because the entire graph must fit on the graph paper. If any component of your graph is not on the graph paper, you will not get the points for either the error bar or for the mean, or whatever component of the graph is not on the graph paper. You will not get credit for it. Okay. It must all be on the graph. That includes you made an error, right? So you're using pen, you make an error. You're like, I'm just going to scratch this whole thing out and I'm going to draw my own graph. Don't do that. You cannot do that. Okay. It must be on the graph paper. So I'm going to tell you a trick. Okay. Use a pencil. Okay. So you take a pencil out, and you will have a pencil because it's multiple choice version, right? So use your pencil and go ahead and graph it with your pencil. Okay. Then if you have more time at the end of the exam, after you've done every other question, go back and do it in pen. If you don't have enough time, it is perfectly fine. We will grade your free response no matter what it is written in. Okay. Pencil, pen, Crayola marker, doesn't matter. Okay. As long as you have written words on a paper that we can read, we will score it. Okay. So we don't prefer pencil. It's hard to read and it sometimes kind of fades in transient, but you can use a pencil. And for your graph, definitely use your pencil. Okay. So use the pencil, make the graph. That way, if you make an error, super easy to just erase that error, right? Okay. So going back to our graph, sorry, y'all, I went on that little tangent right there. So now we have to say, okay, well, this is everything we got points, right? So we correctly plotted our means, we have appropriate labels, unit scaling, and then correctly played, right? Okay. So based on the results, describe the effect of caffeine on each of the following. So if I look here and I go, okay, 10 minutes. So my short term memory, right? I have my mean is going up. So logically, caffeine must be increasing my memory, right? Because look, the mean went up. But notice these error bars. You see how they have the same Y's? So because of the fact that my error bar overlaps, that shows the data is not statistically significant, okay? That shows me that by chance and by chance alone, okay, this is higher, right? It does not mean it always will. Okay, so overall, the answer to, does it have an effect on short term? No, caffeine does not affect your short-term memory. And that got a lot of students because you could see that line went up. You could see that mean was higher. And that's where they're going to get you on the error bars. They want you to be able to understand, like, what does the error bar mean? The error bar is showing your variance in your data. If it overlaps, then that means that the data is not statistically significant. You cannot trust the fact that this is higher. Okay. So then when we look at long-term, we're looking here. Number one, yes, we see the mean goes up. Number two, do the error bars overlap? No, they do not overlap. So caffeine is going to improve or increase the long-term memory. Okay, so I hope that makes sense. It looks like there's a question in the chat. Oh my God, what is happening? Oh, that's because of all the questions that are on my other stuff. I can't follow the questions that are in the little question box because of the fact that all the questions from today's little question in my story, so I can't follow those. But somebody said, Julie says, how do error bars work while graphing? You're just going to do your plus or minus and add it above and add it and subtract it from below your mean. And so it shows the variance of your data. Okay. So going on to part C. I'm going to tell you right now, you're going to be mad. Just go with it. Okay. So part C, design an experiment. So they want you to design an experiment using artificial flowers to investigate potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits to bees. Okay, so you're testing number of floral visits and concentration. They've told you what the testing thing you're looking at is, okay? Identify the null hypothesis, an appropriate control, and then the predicted results. I'll tell you something. You don't have to design an experiment. You didn't have to tell me an experiment. You were supposed to mentally come up with an experiment. There's no points for as much as you want to tell me about this exact experiment. The only three things they're looking for: null hypothesis, control, and then what results would reject your null. You don't have to tell me anything else but those three things. So just take a moment to just let that soak in. Okay, sorry. Nothing I can do about that. I would have written a whole thing, so don't worry, y'all. I think I even did write a whole thing when I wrote the answer to this question, because I always answer the questions when they post them. So first thing, what is a null hypothesis? Null hypothesis says that whatever is happening in your experiment is not going to have a difference between your control and your experimental test. Okay, so in this experiment, we were testing an increase in caffeine, and is there going to be a change in the floral visits? So will we see more visits or less visits to our flower between the control and the experimental? And the answer would be there would be no difference, right? Your control and your experimental results should have the exact same number of floral visits. Increasing caffeine has no effect on the number of floral visits. Floral visits will be the same number between our caffeine and our control. Any of the above is what you should have written. Okay, so the answer that they had was increasing caffeine concentration has no effect on number of floral visits. Notice that it has parentheses, which means you didn't even have to say that. So somebody in the chat says, so why didn't they have to describe the experiment? I can't answer that. I just know that at the reading, I read this question, we were not looking for you to tell me about an experiment. It's bolded. I agree with you. I argued for you, but no, you didn't have to design the experiment. So the second part, an appropriate control. Okay, so the flowers naturally have nectar, and the nectar has caffeine, right? And so if I'm using artificial flowers in my experiment, you can't say I'm going to use a normal flower, because a normal flower, number one, is not artificial and is adding more variables into your experiment. Number two, it has caffeine in it, and we don't know. Well, we could figure out how much caffeine, but we want there to be no caffeine. So what you need to have for your control is an artificial flower with no added caffeine. An artificial flower with no caffeine. You need to somehow connect it that it's an artificial flower and there's no caffeine in it, because I need there to be that baseline of the no experimental component. Okay, so literally writing flowers with no caffeine, nectar with no caffeine, something about that artificial flower. Okay, and then the last thing, we had to come up with results that would reject our null hypothesis. Okay, so our null hypothesis was that, whatever we came up with, whatever the experimental was going to have no effect, right? So we'd have the same amount between my control and my experimental group. Okay, so in terms of your predicted results that would reject this, there is an increase in floral visits in the experimental group. There is a decrease in floral visits. There is a different number, a statistically significant different number. All you had to do was bring in the fact that they were different. Now, you could still get this point if your null hypothesis was wrong, as long as whatever you told me about your predicted was going to reject your null that you came up with. Okay, so they try not to marry the points in which you have to get 1 point to get the next point. They try to make the points independent points where you can get each one on its own without knowing the other parts, which is why you could have analyzed even if you didn't graph your error bars, because you could have added and subtracted and seen that there was going to be an overlap. Okay, is it easier to see with the error bars? Yeah, but you didn't have to. Okay, so as we see there, the answer we look at is that their number of floral visits is different at increasing concentration. Normal floral visits is different than control. So overall, you're showing that we have a difference between the experimental and the control. Okay. And then we on to our part D. This is a longer response, so I'm sorry it's taking me so long to explain this. I'm just trying to make sure we have as much information as we possibly can, right? So researchers found that the nectar with caffeine tends to have a lower sugar content than nectar without caffeine. Plants use less energy to produce caffeine in nectar than they do to provide the sugar in nectar. Propose one benefit to the plants that produce nectar with caffeine and a lower sugar content, and propose one cost to the bees that visit the flowers with that produce nectar with lower sugar content. Okay, so let's think about this. We're going to separate these things out, right? So I need a benefit for the plant, right? So if the plant doesn't have to put in as much energy for to make the sugar and do all that, right? They don't have to put in that energy. They can use that energy for growth. They can use that energy for something else, okay? So you can't just say that they saved energy. You had to tell me what they saved that energy for. Due to less energy put into the nectar, there was a excessive energy that could be used for growth of the plant, for cellular respiration of the plant, for blah, blah, blah. Give me a reason of what they're storing that energy for. And then a cost to the bee, right? So what would be the problem with there being less sugar? Well, if there's less sugar, then my honey's not as good, right? Because now there's a lower sugar content in my honey. Also, I just flew all the way to this flower and I wasted my energy to get to that nectar and there's not enough sugar there. So I'm wasting energy. I'm having to visit more flowers to get enough energy, enough sugar to make my honey. My honey is going to decrease in quality. So all of these are reasons and costs to that bee, and you had to keep them separate. Now, there was a problem. Do you remember? I don't think you remember this, but in the prompt, we mentioned that something was, I think the caffeine was toxic to the bee. That had nothing to do with this. And so don't get distracted by some of these like extra phrases that they'll tell you. They put stuff into the prompts just to give you information. And it's almost like a hook. So be very careful about potentially going down one of those rabbit holes. Okay. Use what you know and apply what you know, but don't fall into the potholes. And it's hard to not know what's the pothole and what's not. I think there was a question that came in. For the hypothesis, do we have to use an if-then? No, you don't have to use if-then statements. Okay, so here's the answers. Okay, so talking about the proposed B cost, talking about the visits, and I think I forgot to put the benefit. I was putting this together quickly. I'm sorry, y'all. Okay, so 2016, number 8. Research conducted an investigation for the effect of exercise on the release of prolactin into blood. Researchers measured the concentration of prolactin in the blood of 8 adult males before T equals 0 hour and after 1 hour, T equals 1 hour of vigorous exercise. As a control, researchers measured the concentration of blood prolactin in the same group of individuals at the same time of day 1 without having the exercise. And the results are shown beautifully here in this graph. So look, another error bars that we're going to talk about. So justify the use of the without exercise treatment, right? So why did I do the exact same thing and like a whole week later and then how I have them exercise? Well, what if naturally at this time of the day you have high prolactin levels? What if it had absolutely nothing to do with the experiment, right? So we need to get your baseline values, right? We need to see, okay, where is the blood normally at, right? Is it possibly high? Is it possibly low? And so we need to be able to use that information. So we wanted to attribute any type of change that you see to the prolactin itself. It can't be due to anything else. Like, so we want the changes in prolactin to be due to the exercise, not due to natural fluctuations in your body, not due to the fact that you might just have a high prolactin level. So it's to rule out those differences. Okay. And so a lot of students get very vague when it says, well, why did you use a control? Okay. So you need to be very specific about the experiment. Okay. And tell them why in that specific experiment was that the control. Okay. So here we see our graph, right? And it says, where's my graph? I must have forgot to give you the question. Sorry, y'all. Anyways, the question is, I believe something about, let me see. Okay, so use evidence from specific treatments to determine whether prolactin release changes after exercise. Okay, so using the results, I need to see, does it actually change, right? Sorry, and I didn't flip the camera just now because I had the answer on the screen. Okay, so if I look here at with exercise, so from time 0 to time 1, right? What do you see? It went up, right? So exercise, of course, is causing my prolactin level to increase. Y'all, are my error bars overlapping again? Dang, they are. Which means that actually exercise does not increase prolactin levels because the data is not statistically significant, right? And so we can also use, that's not how AP exams work. What do you mean that's not how AP exams work? I'm talking about. Okay, so you can also look at, and I couldn't ignore this one, but if we lose these two right here. So if you look here, right, at the 1 hour with exercise and 1 hour without exercise, then of course you're also going to say that we have an increase in our mean, right, with exercise. So logically you wanted to say, yes, it increased it. But again, our error bars are overlapping, which show that the error bars are overlapping, which means, again, not statistically significant. So you could not say that prolactin is going to increase. I'm sorry, that the exercise is increasing the prolactin error bars. Yes, overlapping error bars means no statistical significance. It was due to chance alone that we saw higher levels in the exercise than the no exercise. Okay, so somebody asked the question, I guess, about the way that the exam is scored. Your exam is scored on a cut score, and so they use a whole lot of math to figure out the rigor of the questions. And then they basically compare between your multiple choice and your free response, and they ensure that you're at appropriate levels. And if you are at that appropriate level, then you get a certain score. So they're cut scored. So it's not based on anyone else taking the test. They do find that, I think last year or 2 years ago, it was like 5% got 5s, like 20 something percent got 4s, and like 30 something or 40 something percent got 3s. It ended up, I think that 60% passed it. So I don't remember. I don't focus on the percent that passed because I just care about y'all trying and learning biology. Anyway, so we had to justify our answer, right? And so the problem the students had here was that they weren't specific. They just said error bars overlap. There are 4 error bars here. Which 4 or which 2 overlap with each other, right? Which ones are we looking at? Okay, so you can either talk about these 2, right? Because we mentioned the with exercise. So you specifically had to say that the error bars for time 0 and time 1 with exercise overlap. Or you could have talked about that the mean plus it is within a 2 standard error bars of the mean. And you know that that's what SCX is, right? Standard error of the mean. And then you could have also done looking at these 2, right? So you could have compared time 1 of without exercise and time 1 with exercise. And again, said that those error bars overlapped. Okay, so again, if you're going to say the data is not statistically significant, you need to tell them which error bars are you looking at, and you need to be very, very clear about which ones you're looking at. You can't just say the first one and the fourth one. What are you talking about? Give them details based on the graph. Don't be vague. Tell them exactly what you're trying to let them see. We cannot interpret your responses. Either you wrote it or you didn't write it. If you didn't write it, I can't give you credit for it. It sucks, but that's what it is. Okay. So another question that I graded, which I graded that last one too. So here's another one that I graded on the exam. This one is showing you how you can get points even though you know nothing, right? Okay. Anyway, so this is actually, I think it was like Darwin's nephew, maybe. I can't remember who it was, but he's the big scientist that did this anyways. So phototropism in plants is a response in which a plant shoot grows toward a light source. The results of 5 experimental treatments from classic experiments of phototropism are shown below. So you see here we have an unaltered tip that is going to grow toward the light. We have a tip removed, so they basically took the top off of it. We have a tip covered by opaque cap. So there's this cap up here that's kind of covering this little part right here so that the top doesn't reach the light. Here we've cut the top off and then we reattached it, but there is a permeable barrier. And then here we see that there's a separated tip and then we attached it back with an impermeable membrane. So real quick, because I want to try to answer your questions that are coming in the chat. It says, what if the error bars meet at the very end? Does it still count as not being statistically significant? Trust me, it's going to be obvious. They're not going to put something that's like barely there. Like, oh, it could be there. It could be there. They are not designing the test to trick you. They're not designing the test to be like, got you. Okay. They want to determine, do you know the content or not? Okay. So I would not worry about it being literally at the same error bar, because if there's an overlap, you're going to know there's an overlap. It will be obvious that there's an overlap. Okay. So back to this question. Sorry, y'all. So give support for the claim that the cells located at the tip of the plant detect the light by comparing the results from group 1 with the results of group 2 and 3. Okay, so here let's look at 2. So if I look at 1 and 2, the only difference between 1 and 2 is, look, the tip was cut off. So I have a tip, it grows toward the light. I don't have a tip, it grows straight up. That's the point. Like, literally, tell me the difference, right? If I don't have a cap, the plant grows straight up. Like, that is a point. Like, seriously, that is a point. All you had to do was look at the graph. I mean, the diagram is like, oh, wow. When the tip is removed, it cannot bend toward the light anymore. And that was the point. Now, the problem is that students didn't connect it back to the unaltered tip. So you had to compare the unaltered tip, so tip group 1 to group 2. And then you also had to compare group 1 to group 3. Okay. So now here, I didn't cut off the top. I just covered the top with something that allowed it to not reach the light. Okay. So again, what is the difference? If it's uncovered, it grows toward the light. If it's covered, it grows straight up. Boom. There is our point. So treatment 3, the cap blocks the light from the tip and the plant no longer bends toward the light. It grows straight up. Y'all, we literally just looked at a picture and got 2 points from looking at a picture. Take those points and run with them. Okay. So now the second part is a little bit harder for us to, pull from data. Like, you're going to have to use a little bit of knowledge here. Okay. So in treatments 4 and 5, so over here, the tips of the plants were removed and placed back onto the shoot using a permeable or impermeable barrier. Okay. Well, first thing, what is a permeable barrier? It means the stuff can get through it. What does impermeable mean? Stuff cannot get through it. Describe 2 additional characteristics of the phototropism response. Okay, so I need to have 2 more things that help me here. Okay, so wait. When it's removed, it grows. When it's there and it's removed, it grows. But if it's have an impermeable, it means it cannot grow. Okay, so there must be something in the tip that went down. And because it went down, it allowed it to grow. Okay, so there must be 2 parts. And those 2 parts would be that the tip has some type of substance, a hormone, that's going to allow the plant to bend. You also had to say that the substance must have diffused from the tip lower down. Okay. So hope that was helpful. I know there's a bit of side chatter happening in the chat, which is fine. I think they're all helping each other. If you don't know what the CED is, that is the curriculum exam, course exam description. I've shorthanded it so much that I don't actually remember anymore. If you have no clue what I'm talking about, search AP Bio CED. You will pull it up. It is the exact thing that they're going to test you on. It is all of the content, every single bit of it. Okay. If you look at my big, huge, packet, it's also included in there, I believe. So yeah, there we go. Okay. So the last question we have is about these guppies. Okay. And I did not grade this question on the exam. So adult male guppies exhibit genetically determined spots, while juvenile and adult female lack their spots. In a study of selection, male and female guppies were genetically diverse populations were collected from different mountain streams, placed together in an isolated environment containing no predators. And the studied population was maintained for several generations in the isolated area before being separated into two groups. One group was moved to an artificial pond containing a fish predator, while the second group was moved to an artificial pond containing no predators. The two groups went through several generations in their new environments. At different times during the experiment, the mean number of spots per adult male guppy was determined as shown in the figure. Vertical bars represent 2 standard error bars of the mean. Okay, so here is our graph. Okay, anyways, I'll answer that question in a second. So we need to describe the change in genetic variation. Okay, so as we said before, error bars show you the variance in the data, right? If my data is number of spots, then of course, this is showing you how many kind of different spots those organisms have, right? So between times 0 months and 6 months, how did those error bars change? Well, my SEM got smaller, which means that I had a decrease in variance. Genetic variation is decreasing. Why? The SEM got smaller. You literally just looked at a graph and told me in the graph that we're seeing our genetic variation got smaller, right? Which means that it changed. So all they tested there is, do you understand what an error bar tells you, right? Do you understand that the error bar is showing the variance in the data? Because that's all they were looking at here was, did the variance increase or decrease? Variance got smaller, got decreased, right? And it's because the SEM bar got smaller, right? So the second part of this question, it says, propose one type of mating behavior that could have resulted in the observed change in the number of spots per adult male guppy between 6 and 12 months in the absence of a predator. So when there's no predator, we see an increase in the number of spots. Why would I see an increase in the number of spots? Maybe the more spots they have, the more likely they are to attract a mate. So you could talk about sexual selection, right? You also could talk about that maybe just random mating allowed it to increase the number of spots, right? Just by chance, right? And so it's literally just had to use a little bit of biology and say, well, sexually selected. The ones with more spots were more likely to reproduce. If they're more likely to reproduce, then they're more likely to leave more offspring with more spots. And that's why we see an increase in those frequencies, right? So part C, that propose an evolutionary mechanism that explains the change in average number of spots between 6 months and 20 months in the presence of a predator. So now we have a predator and we're seeing a decrease. Well, what is the evolutionary mechanism there? So I move from having spots to having no spots. Okay. Sorry, I have to turn my camera. I have a big thing against my children being on social media. My son just walked in the room and I can't have him on the social media. Sorry. Okay. So, as you hear him in the background. So as we see, there's been this change, right? So it has moved from, this was almost like 12 spots down to like 9 spots. Well, why? We saw directional selection, right? There's some reason the spots maybe increase the probability that a predator is going to see them. You talk about natural selection because of the fact that they're more likely to be eaten. We're going to see that the ones with more spots more likely to be eaten and less likely to reproduce, and thus they were less likely to pass on that more spots. If they had less spots, more likely to reproduce. All that good jazz were possible things. Okay, so there is no more FRQ Friday. As a quick reminder, the FRQs that you see next week are confidential. Okay, they cannot be released. They cannot be discussed in any way, shape, or form. No memes, no reposting, no sharing, no discussing, nothing. Okay, until they're posted on on AP Central. Usually 48 hours after the exam, they post onto AP Central. When you see them go on, you may have a field day making your memes. Okay, but until they're posted, you cannot do that. The reason why is because I think sometimes they use some of the other exams and they embed them in there. And so you can't discuss them because number one, you can get your score invalidated. Like they will cancel your scores if you're posting memes. They are all over Twitter. They're all over Instagram. They're all over in all these social media accounts, and they will find you and they will cancel your scores. Do not get your score canceled because you want to be funny. Okay. That means no reposting. If someone shares something, you're like, oh, this is funny. Repost, retweet. No, that will get you in trouble too. Do not engage. Literally scroll past it. Okay. 48 hours later, you were perfectly fine. Repost, have your laughs, be cool. Okay. But for 48 hours, there needs to be radio silence in terms of the free responses. The multiple choice will never be released and you can never talk about them. Okay. So before I sign off, there was a question about the time that we should spend for your multiple choice, and it varies between paper versus digital. But if I'm going to go with the maximum, right? So if you're on the digital exam, you need to spend 1.3 minutes per question. I would probably do about a minute for your questions that are by themselves, and then maybe like 2 minutes for your set questions, because you're going to have to keep rereading the prompt and going back and forth between that big long thing. So I'd be like, 1 minute for your short ones and like 2 minutes for your sets. In terms of your free response, since you can't go back on the digital exam, 25 minutes for your long and 10 minutes for your short, that will completely use up all of your time. If you're on the paper exam, I would do 20 minutes for your longs and like 6 or 8 minutes for your shorts, giving you a little bit of time to go back and look at some stuff. There are 60 multiple choice questions and there are 6 free responses, 2 longs and 4 shorts. So remember, you're AP Bio Penguins. You're dressed for success. Good luck. Bye, y'all.